Hello, hi everyone, I'm Sandhya, your botany lecturer. In this video, we are going to study about the cell, the unit of life. Cell. All living organisms are made up of cells. Okay, sometimes some living organisms are made up of single cell, they are called as unicellular. Sometimes they are made up of many number of cells, they are called as multicellular organisms. So we can say the all organisms, living organisms, mainly they're composed of cells and matter. Then this cell is the structural, functional unit of life, or you can say basic and fundamental unit of life. So, living organisms are made up of cells, sometimes unicellular, sometimes multicellular. And this cell, for the first time, it was discovered by Robert Hooke. But whatever cell he discovered was a dead cell, because he discovered a cell in a cock. This cock is a dead one, dead tissue. So whatever he discovered was a dead cell. Then later on, Anton von Leeuwenhoek, he discovered the living cell, the cell in a living condition he has discovered. Then afterwards, Robert Brown discovered the nucleus inside the cell. Then later on, what happened? The discovery of light microscope has been done, then electron microscope. Then what happened? The small details of the cell have been discovered and matter because of the light microscope, electron microscope. The microscopy was very much developed, which led to the discovery of the small details, the inner details, the minute details inside the cell also. So this was about the discovery. Now coming on to the cell theory. Cell theory was first proposed by Sheldon and Schwann. Cell theory was first proposed by Sheldon and Schwann. They said something about the organisms which are made up of cells, that all living organisms are made up of cells, but they didn't explain how the cells were formed. So this formation of cells was explained by Rudolf Virchow, and he explained that the cells are formed from the pre-existing cells. So he said that omnis cellulae cellula. So he explained like this: the cells are going to form from the pre-existing cells. So whatever cell theory which was described by Sheldon and Schwann was again somewhat modified, and said that all living organisms are composed of cells. All living organisms are made up of cells. Organisms are made up of cells and their cellular contents and the cells are going to form from the pre-existing cells. Cells are formed from pre-existing cells. Okay, so first they said something that all living organisms are made up of cells, and Sheldon and Schwann. Later on, they were not able to explain about the formation of cells. So Rudolf Virchow explained about the formation of cells. He said omnis cellula, cells are formed from the pre-existing cells. So these two were combined, and a cell theory was proposed, and having these two postulates. This is about the cell theory. Now coming on to the size, shape of the different types of cells. Cells are of different types. They are not of one type, okay? They are of different types. we are going to study about the size, shape of the cell. The size, shape of the cell. Size, the cells are of different sizes. So they are very small, like mycoplasma, very small like mycoplasma. One of the organism of mycoplasma, PPLO, pleuropneumonia-like organism, is the smallest living organism. Smallest living organism, living cell, we can say, living cell. Then the largest single isolated cell is ostrich egg. Largest single isolated cell is ostrich egg. This is about the size. Shape, there are of different shape. For example, if you take the RBC, they are biconcave and round and biconcave. Then if you take mesophyll cells which are present in the leaves, mesophyll cells, these are round in shape. These are round in shape and oval in shape. Round and oval in shape. Then if you take the tracheids, they are elongated. The tip shape is elongated, very long, like this. Tracheids, these are biconcave. Red blood cells are biconcave, round and biconcave. If you take the mesophyll cells, they are also round and oval, and inside the cytoplasm, the chloroplast will be present like this. But the cells are round and oval, so they vary in their shape and size. The cells vary in their shape and size. They are greatly vary in their shape. They may be disc-like, round, oval, cuboid, columnar, thread-like, irregular, like that. And this is about the size and shape of the cell. Coming on to types of cells. Types of cells is very much important here. There are two types of cells based on the nucleus. Okay. One is prokaryotic cells. Types of cells. Prokaryotic cells and eukaryotic cells. Prokaryotic and eukaryotic cells. What is the difference? The name itself suggests what they are. Karyo means nucleus, pro means earlier, primitive, not well developed. It's a false nucleus. Okay, so here the nucleus is not well developed. It is not well developed. It is a primitive organisms have such type of nucleus. Primitive nucleus or false nucleus. Okay, it is not well developed. Earlier nucleus. Such type of thing is also called as nucleoid, where the nucleus lack the nuclear membrane. The nucleus is going to lack the nuclear membrane. This is the main difference between the prokaryotic and eukaryotic. The nucleus is going to lack the nuclear membrane. The nucleus is not bounded by the double membrane, nuclear membrane. Then cell organelles also. The membrane-bound cell organelles are not present, whereas they are present in eukaryotic. Okay, membrane-bound cell organelles are not present, like Golgi complex, mitochondria, endoplasmic reticulum. All membrane-bound organelles are not present. They are having ribosomes, that is 70S type, and they are having some inclusion bodies. Reserve food materials are stored in the inclusion bodies. These are the mainly present in the prokaryotic cells. In detail we will study. What about the eukaryotic cell? Eukaryotic cells are those cells which are going to have true nucleus. Karyo means nucleus, eu means true. So these are the cells which are having true nucleus. Means the nucleus is going to have a nuclear membrane. The nucleus is bound by nuclear membrane, true nucleus. Then well developed nucleus. That means it possesses the nuclear membrane. Okay, it is advanced cells, madam. Advanced cells, and they're having membrane-bound cell organelles. Membrane-bound cell organelles are present. Cell organelles are present, like for example, Golgi complex, ER, then mitochondria, chloroplast, all membrane-bound cell organelles are present, and these eukaryotic cells also contain ribosomes. Ribosomes, both 70S and 80S are present. Okay, 80S are present. 70S are present in prokaryotic cells, and ribosomes are present in eukaryotic cells. 80S are present. In detail we will study, but generally here also ribosomes are present, but they're 80S. Then here also reserve food materials are also present. Reserve food materials are also present. They are present in different cells and matter, different also parts. Like for example, vacuoles, they store the food materials like that. Okay, so general differences between prokaryotic and eukaryotic cells are these things. Okay, here they know membrane-bound organelles in prokaryotic cells. No membrane-bound cell organelles. Okay, the examples of prokaryotes are nothing but the bacteria, cyanobacteria, etc. Here the eukaryotic cells are nothing but the plant cells, animal cells, etc. Many cells. These are the examples. Now we are going to study in detail about the prokaryotic cell, especially the bacterial structure we are going to study now. Okay, the bacteria is a prokaryotic cell. Prokaryotic cell. Okay, let us draw the diagram now. Sometimes fimbriae also present, like this. Fimbriae. This is cytoplasm. Okay. The innermost layer which is covering the cytoplasm is called as cell membrane, or you can call it as plasma lemma. Plasma lemma, cell envelope, anything like that. Then outer to cell membrane is cell wall. This is the glycocalyx, which can also be called as slime sheath or capsule. If it is loose, slime sheath. If it is tight, it's capsule. Then these are the flagella for locomotion. These are the sex pili or pilus or pili. These are bristle-like structures called as fimbriae. These are surface structures. So this is why it's called as nucleoid, not well-developed nucleus. Okay, this whole thing is nothing but cytoplasm. So inside the cytoplasm, what are present here? The ribosomes are present. They are not membrane bound. Okay, the ribosomes are present. And these are the plasmids. Coming on to these particular structures are called as mesosomes. Okay, this is the general structure of the bacteria or prokaryotic cell. This is the general structure of a typical prokaryotic cell. Okay. So here the genetic material is not enclosed by any nuclear membrane. It is naked. So hence it is called as nucleoid. And also some of the chromosomal units are circular in shape. The circular DNA are present. Single circular DNA, which are called as plasmids. These plasmids are responsible for responsible to give a character of resistance to antibiotics. They also have ribosomes, and some inclusion bodies are also present. Reserve food materials. Inclusion bodies, where they store the food materials. Inclusion bodies. Then these particular nucleoid, the cytoplasm, all these are enclosed in a membrane called as cell membrane or plasmaremma. This is similar to that of the plasma membrane of that of the eukaryotic cell. There we will study in detail about the structure of cell membrane. Then outer to cell membrane is the cell wall. Cell wall is the one which is rigid, and it is going to give shape to the particular cell. It is useful for structural support, and is also going to protect the cell from bursting or collapsing. That is about the cell wall. Then outer to cell wall is the glycocalyx. Glycocalyx is nothing but glycocalyx it is sometimes loose. Hence, when it is loose, it is called as slime layer. If it is tight it is called as capsule. And coming on to the cell membrane, sometimes the chemical composition of the cell membrane is such that then if it is stained with gram stain, if that stain is present, such type of bacteria is called as gram positive bacteria or gram positive. If this particular cell membrane is going to leave that particular gram stain, that is called as gram negative bacteria. So depending on the chemical composition of the cell membrane, they are called as gram positive or gram negative. And this particular cell membrane, cell wall, glycocalyx are going to give the protection to the bacteria. Then there are small invaginations are present from the cell membrane, which is called as mesosomes. These mesosomes, they are helpful in respiration, they are helpful in DNA replication, they are helpful in secretion, they increase the surface area of the absorption, and also these are in the shape of vesicles, tubules, and lamellae. Okay, these are the invagination of the cell membrane. These are in the form of vesicles, tubules, and lamellae. They play an important role in the function of respiration, DNA replication, secretion, increase the surface area of the cell membrane for absorption and enzymatic content like that. So they play an important role. And in some this is about bacteria, no? In some cyanobacteria, chromatophores are present where pigments are present. In cyanobacteria. Then coming on to some of the prokaryotic cells or bacteria are motile. They can move on matter. So this particular motility is due to the surface structures, that is flagella. Flagella is a surface structure which is going to arise from cell wall. It is useful for the motility of the organism. And this particular flagella is going to have the basal body, the hook, and the filament. This is the basal body. This is the hook, parts of flagella. Hook, and this is called as filament. This is about flagella. Single one, flagellum. Plural, flagella, which is useful for locomotion. Then another surface structure which is nothing but pili. Pili is also useful for motility, and sometimes in conjugation also. A conjugation tube will be formed where the transfer of the DNA takes place from one bacteria to another bacteria. So it also forms a conjugation tube. So pili. Coming on to fimbriae. Fimbriae are nothing but they are bristle-like structures. They are going to help the bacteria or organism to attach to the substratum when they're living in water. Okay. These are helpful. These are helpful to attach the bacteria to the rocks in streams and to the host tissues when they're present in the in any host. This is about the general structure of the prokaryotic cell. Then coming on to the ribosomes. Here ribosomes are present, but they are 70S type of ribosomes. There are two subunits in 70S type of ribosomes, 50S ribosomes and 30S ribosomes. And these ribosomes, they may bind to the mRNA like this. They may bind to the mRNA like this. When they bind to the mRNA, they are called as polysomes or polyribosomes, which are helpful in protein synthesis. So the function of ribosomes is nothing but for protein synthesis. They are helpful, and these ribosomes are going to bind to this particular thing is mRNA. When they are binding to mRNA, they form the polysomes and polyribosomes. Why they will bind to mRNA? For the protein synthesis, they'll get bind up. Okay, 70S type of ribosomes are present. Then coming on to the inclusion bodies, the reserve particles are stored in the reserve particles are stored in the cytoplasm, and they are just lying in the cytoplasm in the form of inclusion bodies. They are not bounded by any membrane. They just lie in the cytoplasm. They store the reserve food materials, like for example phosphate granules, Synophasian granules, glycogen granules are the reserve food materials of these prokaryotes. General, they are not bounded by any membrane. They are not bounded by any membrane. Sometimes vacuoles also may be present. Vacuoles are present in cyanobacteria, photosynthetic bacteria, like that, gas vacuoles. This is about the general structure of the structural details of the prokaryotic cell. Thank you.